Hi, I'm Jonah Comstock, Editor-in-Chief of pharmaphorum, and I'm here at the AAPS National Biotech Conference in Boston. I'm joined by Eric Luellen, the founder and CTIO of Turing Biosciences. Thanks for joining me, Eric.
My pleasure, Jonah. Thanks for having me.
So tell me a little bit about Turing Biosciences.
So Turing is focused on a mission. Our mission is around this idea that we believe the historic opportunity for AI is to help reduce the inequalities that are really tearing society apart. And the way that we went about doing that was actually indirect to biology. It was to try and solve a math problem. And the math problem that we were curious about solving is called the combinatorial complexity problem. So if you can imagine DeepMind's AlphaFold is able to predict and really be like a GPS for structural biology. So our question and curiosity was, could we combine different forms of AI and machine learning and apply them to do the same thing for functional biology and systems biology, such that we could create a system that could tell us which molecules, which genes, which proteins, which metabolites were able to immunomodulate any disease and really be a GPS for discovery.
Easier said than done, I'm sure. I know that part of what made AlphaFold work is a really robust available data set. So yes. Where do you find the data you need to do that?
Well, this is one of the core problems. You hit right at the heart of it. We are incredibly grateful to the Nature Publishing Group, which several years ago made it a requirement to post all original raw data if you would like to be published in a Nature journal. So we're able to benefit tremendously from these secondary data sets. One of the things that could have been a weakness and I think ended up being a strength is the multi-omics, because you don't get to define your data set, right? You don't get to design it or pick it. And so often what happens, you say, well, we have a metabolomics data set around this disease, and then we also have some microbiome data, and we have some genomic sequencing data, and so you end up getting this this hodgepodge of data. I have little children, a little daughter. I think of the story Stone Soup.
Yeah.
You put all of the different ingredients into the soup. And so this really was key for us because we wanted to be able to solve that problem. We had to solve the multi-omics. We didn't want to come in with any biases saying, oh, we think it's this gene, or we think it's this microbe, or this enzyme, or this protein. We wanted to come in completely untargeted and say, look, we have all of these data points around this disease. We're not going to assume that we know which one immunomodulates and to what degree. And so if we answer the question right and we solve the math problem, it'll tell us.
Right, because if you go in with that assumption, then you end up biasing the algorithm.
Absolutely.
Right?
Absolutely.
So you've got a vision, an idea, a really interesting idea. How do you turn that into a business? What does Turing Biosciences do in terms of sort of everything you just said, but also making money or staying alive?
Yeah, great. Also a great question. Well, we think that we have, at least in version 1.0, maybe 1.1, we think that we have now solved that foundational historic mathematics problem. We took this system, we've now applied it to 59 diagnostics and therapeutics, new ones that we've discovered in 30 months. We've done that with a very small team of people. We ended up then needing to do exactly what you asked about, which is how do you make this a repeatable, scalable business? For us, that initial choice was to focus on rare diseases. Initially rare pediatric diseases, we'll probably add to that rare tropical diseases, and this is because of the FDA's economic incentive program with priority review vouchers, and also shorter trials, smaller trials, trials against standard of care or historic data for unmet medical needs in many cases. So we now have 16 rare pediatric disease therapeutics submitted to the FDA, 2 initial approvals, 8 more pending.
Wow.
So we're hopeful that at least 1 or 2 of those result in a priority review voucher. But in the meantime, we have all of the common disease discoveries, which are just sitting there. They're just tabled right now. We have a diagnostics for for Parkinson's disease, pre-symptomatic, that's 84% accurate from a skin swab.
Wow.
We have a diagnostic for autism spectrum disorder, which is an infamously difficult thing to diagnose. It's essentially a cluster of behavioral factors. And they're like, yeah, you hit 6 of the 8. We're going to say that you're on the spectrum, right? And that's above 90th percentile accuracy. We have things for colon cancer, for therapeutics, for Crohn's disease, for ulcerative colitis. And so for right now, those therapeutics are parked. And we need to find partners that can take what we've discovered and have the platform the runway to run with it.
All this comes out of this notion of just ingesting a ton of data and letting the algorithm find the connections, right? So you get things that maybe aren't intuitive and nobody would have thought of them on their own, a skin swab for pre-symptomatic Parkinson's, but the data doesn't lie.
Absolutely. Now, there are challenges with the data not lying. We like to say that, we begin where historic pharmaceutical R&D ends, which is we always use human data. And so we know when we discover something, and we always hold some data back to test and to validate that it hasn't seen, that this signal, this biomarker panel, or these metabolites at above a certain threshold will immunomodulate this disease with 98, 99, 97% of the time. And we found that in human data. One of the kind of on the ground challenges, which I'm actually talking about this afternoon, is how do we reconcile that timeline so we can find a new therapeutic or diagnostic panel and then have the system explain the entire disease model. So it's not just the therapeutic that's discovered, it's all of the pathways that it informs and how that fits into a whole model. So it's a little bit like GPS in that it tells you not just where you are, but the fastest way to get someplace. And by the way, here's the model and the framework that you're working within. Right? So we can do that in about 5 days.
Wow.
From start to finish. That's a 2025, 2026 capability. We have a regulatory environment framework that was built really in the 80s and the 90s, a little bit the early 2000s.
You can't possibly keep up with that pace.
Well, so this is a primary challenge, is how do we bridge a framework that was built to handle a technology before that technology even existed?
Yeah, I kind of thought that was what you were getting at when you talked about you're starting out in rare disease where the regulatory apparatus moves a little faster. But I would think even there, it's probably not moving fast enough.
Well, fast enough is an interesting question. I mean, when you're a social impact company, when our primary driver is saving lives, right? So our 16 rare pediatric disease applications impact 500,000 children with unmet medical needs. As a father of 3, this is what drives me. This is what really pushes us, is we could change the quality of life or give a life to 500,000 American children a year. And so if a regulator comes back and says, do you have a mouse model? Mm-hmm. Why would we have a mouse model? We have a human model.
Yeah.
And so, yeah, for sure, there's a lot of conversations around, can you move even faster in the rare disease space? The Office of Orphan Product Development, which is this, separate from CDER and CBER, they're the ones that certify orphans and certify rare pediatric diseases and rare tropical diseases, reports directly up to the commissioner. And they have a very traditional mindset. And so working with them, what we've often found is we have to kind of, it's a little bit of go slow to go fast. It's that, we may point out that we found this with this DARPA-inspired machine learning, but then we actually have to validate it using historic methods.
Right. Which isn't terrible in and of itself when you're working with something so new. You do want to make sure that you're not stumbling on a correlation that isn't really causation.
Absolutely. Yeah, absolutely. There's a lot of goodness that comes out of that. I think in a perfect world, in time, the regulators, both in the United States and Europe, will be able to have the talent bank and the personnel, the capability to evaluate, the differences between machine learning outputs and predictive AI versus something else. It'll always be a combination. It'll always be a melting pot.
Hopefully, the more wins you notch, the more people are being inclined to trust this new process.
We think that's true. We've been in stealth mode for 30 months. We're just about to de-stealth. We have a major paper that we're writing that will hopefully be in a high impact journal coming to you soon, coming to a high impact journal close to you soon, which we cross-validate not only with in vitro evidence, but across different modalities, disease types, functional genomics, immunometabolism, and 29 different diseases. So hopefully that does a lot with statistical measures, et cetera. But we like to believe that there will be some momentum. And once we get a few more approved or through the final approvals, that it'll help make those kiddos who have other diseases get those therapeutics faster.
I'm sort of taking all this at face value, but I'm sure when you pitch this, when you're looking for partners and things like that, I imagine there's a lot of skepticism because that can't possibly work. You can't possibly be finding things that quickly or getting that complete information. I mean, how do you respond to that kind of, skepticism?
Yeah, great question. I mean, I think people distrust what they don't understand, right? They trust what they do understand. So there's an educational component. If you have a proprietary AI method, it's hard to get too far down the education road without disclosing to everyone and losing your IP about what you actually do. One of the ways we do it is we've done some studies around retrospective, prospective. So if you take something like colorectal cancer, there's a lot of research out there on colorectal cancer in the literature. And so we took a lot of those data sets, put them through the system, and we were able to juxtapose, this is what the 17 leading studies think they found around colorectal cancer. Here are the immunomodulators. Here's the sequential importance of them in terms of quantifying their impact. Here's which ones are microbes and which ones are metabolites. And here's what the disease model looks like. And the system was able to validate some of those and saying, yes, we found those too, which is a little bit of a toehold in the belief system, if you will. And by the way, here are the missing pieces that no one's found. And so the fact that we can not just discover these immunomodulators, but put them into a disease model and put that into a visualization, so people have spent their careers studying these diseases can look at it and really validate it with their own knowledge. Look at it and say, oh, that's what was happening over there. We never knew what happened over there or how this process works. Yeah. But we understand pathways, we understand what these molecules do. And so this kind of finishes the piece of the puzzle. Puzzle is a good analogy. It's like if you have a puzzle put together, but you're missing maybe 30 or 40% of the pieces, you have an inkling what the picture is, right? You kind of get the gist of it. You're not exactly sure. But when the pieces come into place, you know it's correct because it completes the picture and it aligns with people's biological understandings.
You mentioned that your data set is kind of the raw data from published studies. So I guess that also means that you're bringing along all the baggage that our research publication culture has, right? Yes. The bias against publishing negative results, yes. And other kind of more problematic systemic biases in terms of who gets included in research. I mean, how do you manage all of that and try to make sure that you're not getting played by the data itself?
Yeah, that's a great question. I mean, so a couple ways. One, nature is pretty high standard to start with.
I think everyone would agree with that.
Yeah, exactly. And so we're not having to dip into the low impact score journals where there might be more reproducibility problems or more quality problems. So fortunately, we have a positive bias towards quality by having a lot of data sets that come out of nature. In other words, there are some machine learning techniques that we can do in terms of making sure we're averaging models. The system needs to be able to find the same thing consistently in different methods. And so that's a quality control component. We always reserve 30% of the data just off the top, randomly selected, and set it aside. So
you can check against it.
That's right. We need to be able to reproduce the results 3 times from these journals. As we move forward, quality is a big issue. Your comment about negative results, I think, is just so important because one of the things the system does is in the earlier steps when it discovers the immunomodulators, before it puts the whole disease model together, it'll tell us in ordinal ranking to the hundredths of a decimal the impact that a molecule has on a disease state or a phenotype. right? And so what often ends up happening, which is very interesting, is that let's say there's 1,000 variables that we look at. It's almost always less than 10 that get you above 90% predictability. Literally less than 10. The other 990, I think, is a latent source of tremendous value, almost like a negative results database, right?
Mm-hmm.
That even if we can't tell you until the FDA approves it, the things that are most important, we can tell you 900 things that are not relevant in this phenotype or this disease, which I think is its own way of introducing efficiency.
Definitely, yeah, valuable. What haven't I asked about that you think is super interesting or exciting about the work you're doing?
I think one of the things that's very interesting is there's a lot of homogenous thought around AI that we see right now. And people are understandably biased towards what they know, which is probably consumer things like ChatGPT, OpenAI, Gemini, Copilot, right? And so I think when we're working with regulators, when we're working with decision makers who may be general management and not scientific staff, they look at it and say, well, I don't understand how, I've used ChatGPT. I know what AI is, right? I use it all the time. I ask it to look things up for me, wrote my bio for me. I don't understand how that could find a drug. I don't believe that.
Yeah.
Sometimes it says things that are true. And it turns out that predictive AI really is in our experience, is a very high risk approach today to do biological discovery. But the machine learning side, which is much more statistical driven, is also more complicated to explain. So you end up with this a little bit of a conundrum. There's the AI that people know they don't have high confidence in, and the AI types of AI that they should have confidence in, there's a knowledge gap that there's not a reason for them to be familiar with the inner workings. And so we end up having to do a lot of thought models when we talk to people and explain things, and trying to find the test statistics that they understand. And if the system's output is correct, all the test statistics are good, right? So specificity is good, sensitivity is good, total accuracy, good old confusion matrices, right? Area under ROC curves is good. And so that is a primary challenge. At the end of the day, I think the final validation is really via traditional methods and seeing human results, interventional studies as opposed to retrospective studies. Yeah.
Well, I wish we could keep talking about this. It's really fun, but I think
It
probably
is fun.
this is already far too long a
Jonah.
video. So thank you so much, Eric, for
My
chatting.
pleasure.
I'm really looking forward to seeing what comes out of your guys' shop.
Thank you. Thank you for having me today.